Hello and welcome back to our YouTube channel. This video is in continuation with the previous lecture where we have already discussed the initiation of pre-fertilization events in flowering plants with the changes in hormones and structure that leads to development of flowers. Flowers bear androecium and gynoecium, that is the male and female reproductive organs. In this video, we'll be focusing on the male reproductive organs and the pre-fertilization events taking place in them. Stamen structurally is consisting of a terminal anther and a stalk-like filament that is attached to thalamus or petals of a flower. Stamens may be of two types: monothecous with one lobe and two pollen chambers, or dithecous with two lobes and four pollen chambers. This image here shows a green colored filament that has anther at the terminus. Anther contains microsporangia, that is the site for development of pollen grains. If we take example of a bilobed or dithecous anther to understand rest of the processes taking place in them, a bilobed anther will have tetragonal structure with four sides or four corners containing four microsporangia. There will be a total of two microsporangia in each lobe. Microsporangia develops into pollen sac and contains pollen grain. Considering the transverse section of microsporangium, there are four layers in its wall. The first three layers, epidermis, endothecium, and middle layers, these are for protection and for dehiscence of anther in order to release pollen grains. The innermost layer, tapetum, is required to provide nourishment to the developing pollen. This image shows a tetragonal anther containing four microsporangia located at each corner of this tetragonal structure. In other words, each lobe of the anther will contain two microsporangia. Now, apart from other layers of microsporangial wall, each of these microsporangia will contain uh, a group of compact, dense sporogenous tissues when the anther is young. In the due course of maturation, each of these sporogenous tissue is a potential microspore mother cell or pollen mother cell, which can divide by meiosis and give rise to a group of four cells known as microspore tetrad. These microspore tetrad can further mature and dehydrate into microspores, which will dissociate from each other and develop into pollen grain. A mature anther will contain several thousands of microspores or pollen grains, and with the dehiscence of anther, they'll be released in order to continue the process of fertilization. Let us learn some facts about pollen grains. These are male gametophytes, spherical in shape and about 25 to 50 micron in diameter. The wall covering pollen grain is known as sporoderm and is made up of two layers, the outer exine and inner entine. Exine is hard and made up of sporopollenin, which is one of the most resistant organic material known to us. It is resistant to strong acid and alkali and even to the high temperature. Exine is non-continuous and has pores in it. If the apertures have circular outline, we call them germ pores, and if apertures are elongated, we call them germinal furrows. The wide array of pattern and design exhibited by exine is shown here in the scanning electron microscopic images of pollen grain, showing how beautiful they look. Entine, on the other hand, is thin and elastic, made up of pectin and cellulose. There are no germ pores, hence the entine layer is continuous. As I have mentioned earlier, during the process of microsporogenesis, a microspore mother cell will divide by meiosis into microspore tetrad, and each of this microspore tetrad cell will independently mature and develop into pollen grains. Now, each of this pollen grain undergoes maturation by undergoing mitosis, where asymmetrical spindle formation leads to unequal division of the cell and gives rise to two cells: a bigger vegetative cell with abundant food reserve and a large nucleus, and a small generative cell with dense cytoplasm. This is known as two-celled stage of a pollen grain. And in almost 60% of angiosperms, the pollen grain is shed at this two-celled stage. Generative nucleus is supposed to divide into two male gametes through mitosis. Once there is formation of two male gametes, the resultant structure is known as three-celled stage. So in 40% of the angiosperm, the release of pollen grain is in three-celled stage. You might have heard people getting sick when they come in contact with pollen of certain species. Pollen allergy may lead to runny nose, stuffy nose, sneezing, asthma, hay fever, or bronchitis. Although pollen grains are known to cause severe allergies, but because of them being rich in nutrients, there is increasing demand of pollen products in the form of tablets, syrups, and food supplements. Viability of pollen grains depend upon several factors, such as humidity, temperature, or the species of plant. They may be viable for as less as 30 minutes in case of wheat and rice, or up to certain months in case of plants belonging to the family of Rosaceae, Fabaceae, and Solanaceae. That is all for this video. In next lecture, we'll be talking about the pre-fertilization events taking place in female reproductive parts, that is carpel. Thank you.